Can you try again, perhaps? Yeah, perfect.
Okay, great, thank you. So, hello everybody, my name is Marcin and I'm a PhD student at Newcastle University. And today I want to share with you what's bioprinting and introduce you to a bioprinting technology I am working with in my lab, and give you a brief intro how I've been using this technology by exploring its potential for some biomedical applications. So let's start with bioprinting. So basically, bioprinting is a biological cousin of 3D printing, and it relies on a selective deposition of materials in a layer-by-layer manner to build 3D physical objects. And the main difference between bioprinting and 3D printing is, however, that materials that are used for bioprinting are special, because these are living materials, and we call these biomaterials bioinks. And the bulk of many bioinks are water-rich polymers that are called hydrogels. And these hydrogels, they create a friendly environment for a key element of each bioink, cells. And by far, the most popular bioprinting technique for fabricating 3D hydrogel-based structures is the one based on the extrusion, where you have a continuous strand of a gel that is dispensed from a syringe nozzle. So I would say you can think about it like of squeezing toothpaste. You apply some pressure, force, and then you get your toothpaste ready. And the good thing about the extrusion is that it's compatible with many bioinks, but the issue is that when you apply that pressure to get the highly viscous gel out of the nozzle, there are some shear stresses created, and they may negatively affect cells or even destroy them. So another alternative is droplet-based bioprinting technique. And in this one, you dispense a bioink in a form of tiny liquid droplets very quickly. So you can think about this one like applying eye drops. And in fact, it is easier to load liquid bioinks in the form of droplets rather than already viscous gels to the printing head. And also having cells in a low viscosity bioinks puts a lot less stresses on them. But on the other hand, if you want to get this hydrogel friendly environment for your cells and build a 3D structure, you need to think about how quickly you can, after bioprinting, gelify your droplets to achieve a hydrogel based structure. And one of the solutions for that, so you print bioinks as droplets, but ultimately you produce hydrogels, is the bioprinting process I'm working with, called Reactive Jet Impingement, or just REGI. So basically think about it this way, that you are using different eye drops, and you deposit eye drops at the same time. They meet, they mix, and this is where some chemistry, so the gelling reaction happens, and they create toothpaste. So this is basically the idea behind the REGI bioprinting, and this is more scientifically how our bioprinting setup looks like. We load liquid bioinks to reservoirs, and we eject them in a form of droplets through dispensers that are called microvalves. And these microvalves are directed to jet bioinks droplets at one another in mid-air, where those droplets react and form a hydrogel, which then lands on a biomaterial substrate. And basically, this biomaterial substrate acts as a paper for printing. So we can refer to it as a biopaper, as we're using it for bioprinting. And in fact, biopapers are a core part of my PhD project, because we believe that using the Bioprinting, we can bioprint cell-laden hydrogels onto different biopapers that are made of different materials such as metals, bioceramics, polymers. And apart from different materials, we explore also how biopapers characteristics, like for instance texture or landscape, play a role in the process. And putting it in the context of specific medical applications, we investigate how does it influence the cell response. So how we imagine this could be applied. One of the examples is in clinic bioprinting. So using the Bioprinting to either enhance the integration of current implants in the human body or develop new personalized tissue engineered implants. So during surgical procedure, imagine that we could harvest patient cells, process them then to create a bioink, load to a bioprinter, and then the implant. So our biopaper can be integrated with cell-laden gel to create living structure that would get implanted to a patient during the same surgical procedure. And some of the exemplary applications could be, for example, for orthopedic implants or wound healing. And as a part of my PhD, I explore use of the Bioprinting for development of living wound dressing. And we envision that they might have potential to accelerate healing of hard, chronic wounds. And in this experiment, I bioprinted skin cells that are called fibroblasts in a hydrogel onto fiber-based biopaper made of natural polymer. And as a big part of my research is visualization of cells in vitro at different stages to see the cell behavior, or how they respond to the environment, how do they interact with the biopaper. And for that, I use different visualization techniques, for instance, confocal microscopy, that in different colors shows us different bits, let's say, in cells, and gives us information about their shape and their communication between each other. And in gray, scanning electron microscopy that allows us also to visualize our biopaper and study how cells are interacting with the biopaper. Another idea is actually to use the Bioprinting to enhance metal implants with patient cells to make the implants more bioactive. And in this experiment, we investigated the response of bone marrow stem cells to metal biopapers with different surface characteristics. And we checked at different time points the interaction between cells and the implant. For instance, if the stem cells developed into mature bone, and how the cells organize depending on the biopaper landscape and day of experiment using 3D confocal projections. Finally, another great research avenue of bioprinting are in vitro models. So models that are produced in the lab and can be used for drug evaluation and studying various disease progression, for instance, cancer. Having that would faithfully mimic human tissues could help us to eliminate the need for the use of animals for testing. And I don't know if anybody's seen this short animated movie released recently. Actually, it was very popular around social media, of the harshness that animals use for cosmetic testing experience. And I think it's great that society starts to understand that it's unacceptable to make the animals suffer in the name of the human beauty. but the long-term goal would be also working towards eliminating animals use from the drug industry, and that is an amazing tool to do it. So just to quickly wrap it up, hopefully you got a bit of understanding how bioprinting works, and especially the regi bioprinting, and how using it with different biopapers can open many interesting routes for various applications in medicine. I just want to emphasize that bioprinting technology is still in the early stage of development, and researchers all around the world are still learning and addressing multiple challenges, and likely first bioprinted in vitro models may hit the market in more or less 5 years, but ultimately we'll have to wait longer for tissue engineered implants and human organs that we aim to implant in humans. So that's all what I want to share with you, and yeah, thank you for listening.
Thank you, Marcin, for your talk. It was really great. And again, I have a lot of questions, but we also have questions from our attendees. And the first one is Jolanta Palowska. And Jolanta, you can ask your question. Sorry. Jolanta, would you like to still answer your question? Because
Sorry.
Yes.
Sorry, I don't know if you can hear me because I'm having some technical issues. Can you hear me still?
Yes, we can hear you.
Okay, great, thank you.
So it was a mistake on the side of Jolanta, but we have quite a lot of other questions as well. So one coming from Magda Szczygieł. Magda, can you?
Yes, sorry, I had to unmute myself. Great talk, thank you very much. I actually do something a bit similar because I work on similar gels, but actually the cells produce it themselves. So just a quick question: do you? Maybe I missed it. Like your sort of biopaper, how you call it? Is it just artificial polymers, just functionalized with some small peptides? Like sort of recalling the ligands for some cell receptors, or is it actually ECM proteins like collagens, fibronectins, or like sort of natural products which would usually be also, for example, in the skin? Hello.
Marcin, are you with us? Oh, it might be the case that we have some technical difficulties on the side of Marcin.
Sorry, I'm back. I think I was kicked out somehow. Sorry, so I didn't hear. My apologies.
Okay, I was just asking whether this biopaper is some artificial polymer with just small peptides of ECM proteins, or you actually use natural, for example, collagens or fibronectin or other proteins which would also naturally exist as this like scaffold around cells.
Yeah, okay, thanks for that question. So it really depends on the application. We use different biopapers for wound healing. These were natural polymers made of calcium alginate, for example, that is popular material for wound healing. By itself, even our BioInk we are printing is made of proteins like collagen and fibrinogen that are naturally existing in a human body. But for example, for orthopedic applications where you use metal implants, yeah, this is a synthetic metal, pure grade titanium that is used also commercially for operations and surgeries.
And did you, for example, check whether treating, like if you seed fibroblasts in vitro on these different biomaterials, do you see different responses depending on whether you seed them on fibronectin or like fibrinogen or collagen or other things, or they are more or less the same?
No, we clearly see many differences. Like I just couldn't show all the differences and explain how is it going, but depending on the material, depending on the landscape, texture, and also depending on the cells, because I just mentioned that we used fibroblasts for wound healing, but I work with many types of cells and even cell densities. So yeah, we see clear differences.
We also have time for one quick question from the audience. So Alicia, now I will unmute you.
Okay, hey, thanks very much for an interesting talk, really fascinating. I have a very quick, actually, curiosity question, because you mentioned you work with wound healing and fibroblasts, and I'm a neuroscientist, so obviously my mind goes straight there. Do you know that, are there any realistic attempts at using biopolymers in the cells of the nervous system? Because those are slightly more complicated than fibroblasts, right?
Yeah, thanks for that question. That's the main message, more or less, I wanted to communicate at the end of my talk. So bioprinting is at the very stage of development. So researchers are trying to now replicate simpler tissues, and skin is relatively simple tissue. Bone is relatively simple tissue because they are made of layers. But when it comes to nerves, when it comes to heart, there are some very simple attempts. But moving the research from from the lab to somewhere else, it will take a lot of time. So of course there's the literature, there are people exploring, but we need time for that.
Of course, thank you very much. It's something very exciting to look forward to.
It is, yeah, it is indeed exciting. Yeah.
Thanks a lot.
Thank you.
This is exactly the question that I wanted to ask. So I'm